It's Hao, and I'm a final year PhD student at Tufts University. And today I'm very excited to introduce you to RegDiffusion, which is a very fast GR inference algorithm for single-cell data. And as you know, gene-regulated networks, or GRN, is a very useful tool for us to understand the underlying mechanism for many biological processes. It's also very important for us to identify possible targets for the treatment of certain disease. There are many ways to get to a GRN, but in this talk, we focus specifically on inferring GRN based on gene expression data. In fact, this has been an active research field in the past 20 or 30 years. And people have proposed so many different inference methods, ranging from Bayesian networks to decision trees. And in recent years, people are using deep learning methods to speed up this entire process. On the other side, we also see a lot of advancements in the transcriptomics techniques. In fact, one of the challenges we are facing today is that these new transcriptomic techniques, especially with single-cell RNA-Seq, it gives us access to a lot of data in a lot of cells but our computational tools, they don't really scale that well. Among all the deep learning methods, this kind of autoencoder-based structure equation model, it draws a lot of attention in recent years. A good example is called DeepSam. People find that it outperforms a lot of previous methods based on benchmarking results, and it also runs much faster. Last year, our lab also proposed an improved version of DeepSam, and we call it DESO. In this method, we add a small amount of extra zeros to the data, and we find that it actually helps the model to stay more robust. We actually have a poster on this. If you are interested, come stop by in the poster session tomorrow, and we can discuss more. Either way, in all these kind of autoencoder-based models, as you can see, we always parameterize the adjacency matrix of the GRN. As you can see in the figure, it's a yellow box. And we use that on both sides of the autoencoder. Before I move on, I would like to point your attention to this highlighted area where we apply the matrix inversion on the adjacency matrix. As you may know, matrix inversion is usually done by Gaussian elimination, and it runs in cubic time. That says, given any single-cell data with n number of cells and m number of genes, the runtime of all these autoencoder-based methods will at least be n times m to the power of 3. In this talk, we're going to show you how we managed to reduce the time complexity all the way down to m squared in RegDiffusion. So before I talk about the details of RegDiffusion, I would like to give you some background information. RegDiffusion is a class of the denoising diffusion probabilistic model, or DDPM, or sometimes people just simply call it diffusion models. You probably have seen those image generation models with some text input, right? A lot of such kind of product, they are actually powered by diffusion models. In diffusion models, there are two processes. In the forward process, there are no parameters. We gradually add a small amount of Gaussian noise into the cat image until it's going completely noisy. And in the reverse model, this is actually where the modeling piece is happening. So the reverse model takes at least two inputs. The noisy image, of course, we need the noisy image. And at the same time, we need to input the diffusion time step t, which tells the model how much noise should we expect there is in the input data. So the reverse model will be trained to predict the added noise, so that if we want to get a denoised version of the image, we can simply subtracting the predicted noise from the input image. In computer vision, this step is usually done by this kind of U-shaped neural nets called it's just called UNet. But how can we use this kind of model on single-cell data, which obviously they're not images? This is, in fact, the most important slides of this talk. So RegDiffusion is based on this linear additive assumption that is, in fact, widely used in a lot of Bayesian network methods. So let's say we have 4 genes here, A, B, C, and D. And we use the small x to denote the log form of the probability of seeing each gene, okay? And here, the linear additive assumption simply says that XA will equal to the weighted sum of XB, C, and D. This might still be a little bit complicated, but if we write that in the matrix multiplication form, we can simply write that in this very simple version, X equals to XA, where this capital X is a gene expression table. This capital A is the agency matrix. Based on this assumption, we further ask this question: what will happen to this equation if we add a small amount of noise as all these diffusion models, small amount of Gaussian noise? We did a mathematical proof, and we're very happy to discuss it either in the Q&A or after this talk. But here is a conclusion. If we use X prime to denote the noisy version of the data, the X prime A will be a close approximation of the unperturbed X. Given this conclusion, recall that in the diffusion model, we need to train a reverse model to predict the added noise. So with this conclusion, we can simply get this formula saying that, okay, the added noise will simply equal to X prime minus X prime A, or in other words, X prime times I minus A. The I is the identity matrix. So as you can see, this equation is very streamlined. The benefits of doing this is like, right now, there is no need to do the very costly matrix inversion anymore. Here's an overview of the RegDiffusion workflow. As you can see, we start with just simply with gene expression data. The expression data, after we pre-process it, it feeds into the forward process and generate the perturbed XT in the gray box. We send the XT and the diffusion time step T, as I said, into the diffusion model, and that generates the prediction of the added noise Z. Here is a detailed look of what exactly does it look like in the diffusion model. It looks a little in the RegDiffusion model. It looks a little bit complicated, but it's in fact, as I said, it's very streamlined. We always start with a feature embedding layer that transforms the input data into some kind of vector representation. Then these vectors get sent into some layer of multi-layer perceptron to build a better representation. We multiply this better representation with an I minus A matrix, as you saw in previous slides. Finally, just a very simple linear prediction layer to predict the added noise. So it's very streamlined. Okay, now we know the architecture. How does RegDiffusion perform? We tested on the B-line benchmark, which include 7 different single-cell data sets and 3 different sets of ground truth networks. We also tested on 2 subsets of published microglia data sets. So in this, these 2 microglia data sets, they have, after we pre-process it, they have more than 14,000 genes and thousands of cells. So they are more closer to the kind of single-cell data you see in your daily life. Based on our validation, RegDiffusion does great on its performance, but what really excited us is its improvement on runtime. As you can see across all B-line benchmarks, RegDiffusion can almost always finish the inference within 10 seconds GPU computing time. And note that I'm listing the size of the network down below. They're not small networks. They're with like 1,000 genes. In contrast, DeepSam, the ensemble version of the DeepSam takes roughly like 5 minutes, and the Genie 3 will take a little bit less than 3 hours. And this improvement becomes more obvious when we have more genes. So the ensemble version of the DeepSam, it takes more than 40-hour GPU computing time. While you probably can't see it on this figure, the yellow box of RegDiffusion on this figure because it's too small. The RegDiffusion only takes 3 minutes. The significant speed gain of RegDiffusion not only comes from the elimination of the matrix inversion, but it's also due to the fact that we are using a fixed size bootstrap sampling strategy. So during the training process, in each step, we sample a fixed amount of data and feed that into the model. By observing the amount of change in the gene sequencing matrix, we realized that, okay, so regardless of the size of the data, the amount of change in the gene sequencing matrix throughout the training process will always follow a similar pattern. So we conclude that in this case, with this set of hyperparameter, our model will always converge roughly after 1,000 steps. Okay, as I said, RegDiffusion does great on the benchmarking results. Here are two tables with two different metrics, and RegDiffusion is the one listed at the bottom. You probably can't read the numbers, but you can easily identify that RegDiffusion, in most cases, is either the top performer or the second top performer across all the methods. We have similar findings on other based on other ground truth data, and all the results are provided in the preprint in the paper. Okay, so we in addition to all these benchmarking results, we also want to show you what the actual inferred networks looks like. In this case, like remember that for the microglia data, we have more than 14,000 genes. We don't want to show you a big hairball with 14,000 genes. That's too much. Instead, we decided to show you what you see here is like it's a two-hop neighborhood around particular interesting gene, which in this case is ApoE, a very popular Alzheimer's disease gene. And we also use colored ovals to denote the functional groups that we find are linked to ApoE based on existing literature. As you can see, they are very sensitive. And another interesting finding that excited us is that we find that across these two data sets, it's a Hamon data set and Atlas data set, although they come from two different groups, they are collected from two slightly different regions from the brain. And the Atlas data is filtered by marker genes, while the Hamon data set is filtered by experimental methods and include more subcell types. Despite all these differences, we can still find a coherent regulatory region as noted by the gray box. And before I move on, I would like to point your attention to this green oval, the cathepsin group. Cathepsin is a group of proteins whose purpose is to break down proteins. So it makes sense that it's interacting with a core group of genes in microglia, which is one of those immune cells. And fortunately, we also find cathepsin gene existing in other B-line benchmarks, in those benchmark data. And we find that the inferred network around cathepsin, in this case, cathepsin D, is in fact very specific to the cell type. For example, in the mouse embryonic stem cells, cathepsin D is directly linked to BTG1 and 2. They are anti-proliferation factors. In the embryonic stem cell setting, they are acting as regularizer after all the growing factors, which totally makes sense. Okay, finally, it's a conclusion. RegDiffusion is a very fast algorithm, and it works great. The network generating is very trustworthy. And also, as one of the first diffusion models in this field, we think it has a great potential in single-cell analysis and in network inference. Okay, so finally, it's supposed to say software. Finally, because we are confident with these methods, and we want to make sure people have the ability to actually use it, we put in quite some efforts into developing this software. It's available on GitHub and Pipe. Here's a quick example on how to use it. You can quickly start a trainer, simply train it with one single line of code. After you train it, you can export the GRN as an object, save it as a HDF5, and you can also visualize local neighborhood, generating some figures you just saw using one single line of function. Finally, it's a group picture of Tufts PCB group, which is very active and supportive. And I'm also looking for postdoc positions as I'm graduating. And finally, I also want to take this opportunity to thank my mentor, Donna Stonian, sitting down here. He's always been so supportive and helpful. If you are interested in this topic, in this talk, here are the QR code for the GitHub page and preprint, and I'm ready for questions.
Questions about the talk, not about an interview for postdoc position. It's not about So I had one. I can start. I was wondering, because something that is often quite difficult is to plug a single-cell data set coming from different experiments. And do you think your architecture, you could plug like your different latent space or something at one point?
Yes. In this case, we have two kinds of latent space, or like trained embeddings. So first of all, we have in the network, you probably noticed that we have a gene embedding layer. So right now, we train a new network on every single data. So it's not really meaningful, but I think after we train again, maybe we can generate a more representative representation for each gene. And at the same time, what's coherent, what we hope that can stay across data set is the adjacency matrix, or the interaction among different genes. And just imagine that later in the future, if we apply this on a lot of data and we build a coherent adjacency matrix under different conditions, that will actually be helpful. Yeah.
Yeah, even different conditions, different types of omics also could be interesting. And
exactly.
Okay.
Hi, really great talk. So I'm wondering, I think that the error objective that you give is related to a causal structural equation model.
Yeah.
So first, like, is it mandatory that the graph be, a DAG for this? And then if it's a DAG, do you make comparisons against something like the no tiers method of
yes.
DAG recovery?
So right now we are following the setup of DeepSam, and we are not applying any DAG restraint. So in theory, because DeepSam is pretty much like a DAG GN without the DAG part.
Mm-hmm.
So you can add a loss function for the DAG for the community, but you need to change the optimizer.
Okay. Yeah. So none of the evaluations are constrained to be the DAGs in that sense. Okay.
No.
Thank you.
And also, I mean, a lot of these kind of gene regulation, there are a lot of loops, so it's not a very strict DAG.